Okay, ANP2 students, this video is going to talk about the urinary system as it pertains to the waste products. And there's 3 of them that we'll talk about. And then we'll talk about the processes that are going on in the kidneys and why they're happening too. And then I'll probably do a separate video for the renal calculi. Okay, so I'm going to find our picture here. Okay, we don't have a picture for these, but these are the 3 waste products, nitrogenous waste products, just meaning they contain nitrogen, that you should be aware of. I did, as I was going through these, I kind of changed the wording of the objectives, and I'll put this, update this objective list on Blackboard, just because I noticed that it wasn't really as clear as what I intended it to be for what you need to know. Same way with 26, objective 26 as well. So you can identify the source of those 3 nitrogenous waste products. So the first one is urea, and it's a byproduct of the breakdown of protein in the liver. So that's where that waste product is coming from in the body. And then the uric acid, it's also a byproduct of, in this case, it's the breakdown of nucleic acids, and that also occurs in the liver as well. And then the last waste product is called creatinine, and that is produced, and it's a byproduct of the breakdown of creatine, which is in our muscle tissue. So that's where those 3 waste products are being formed. And they're things that we want to get out of the body. We don't want them to be, they have normal ranges in the blood, just like other ions and hormones and things. But they're going to be circulating in our blood because we have a constant breakdown of proteins and nucleic acids and the creatine in muscles. But we don't like those to get any higher than that. That usually indicates there's an issue going on with our kidneys. So the other part of the objective is to just understand their elimination by the kidneys. And so when it comes to urea and uric acid, they get filtered. So realize all 3 will be filtered. But the urea and uric acid, as they move along the nephron, they're going to be reabsorbed and secreted. And it'll vary depending on the which one we're talking about. And realize too, urea has a role in the kidneys. So even though it's a waste product, the kidney also uses it for a purpose. Not all of it gets into the urine. Some of it will stay within the interstitial fluid of the kidney and help help establish a concentration gradient that we'll talk about in a little bit. So, and then the creatinine is different than these 2 waste products because it's only secreted. All of it gets filtered, and it's almost all of it, and then some of it will be secreted. And then when when we're trying to determine if there's a lot of different ways you can determine kidney function as far as if the kidneys are healthy and working properly. And one of the ways is to look at blood work and look at these waste products in the blood and see what the levels are, and that along with other information may help you determine the function of those kidneys, or if they're not functioning well, at what level are they not functioning. So there's different stages of kidney disease and things like that. So, all right. So that's our waste products, what you should know about them. 26 is in this case, I don't want you to explain counter-current multiplier or counter-current exchange system, because that gets into a lot of details I don't think is necessary. But I do think it's important to know that the purpose of those 2 processes that are going on within the kidneys, and the countercurrent multipliers going on within the nephron, and the countercurrent exchange systems going on in the vasa recta. So that's those peritubular capillaries. And it's mainly the juxtamedullary nephrons, the ones we talked about it in an earlier objective that are the ones that have this counter current multiplier. So I don't want you to know the details, you know, like what's going on and why is this and that, and which way is the water and solutes going. I just want you to realize the reason that the nephron, actually the loop of Henle is actually where the countercurrent multiplier is happening at, and then the vasa recta. Just want you to know that the things that are going on, these two processes, are there to establish a concentration gradient. So what that means is, and you can kind of see over here, in this is the medulla down here. This would be closer to the cortex up here. So we have like more concentrated interstitial fluid down here. So that's where we want this concentration gradient to be. So you'll notice that there's less concentration of solutes, mainly sodium and chloride, which is salt, up here. And then as we go deeper into the medulla, get this darker blue color, that's indicating that there's a lot more salt in there. So a higher salt concentration. And that'll be, I mean, they're showing the interstitial fluid just around the loop of Henle, but realize that'll be all the interstitial fluid in the cortex in the medulla of our kidneys. So even though they're just showing it along the loop of Henle, they're mainly showing the loop of Henle because that's what's establishing this concentration gradient. So, and the concentration gradient is low concentration here and higher concentration here in the medulla area. So what that means is we have to have this concentration gradient where we have a higher concentration of salt here. So that way the antidiuretic hormone that we talked about when we talked about water reabsorption can work. Because if the antidiuretic hormone comes down, and remember antidiuretic hormone is going to cause more water reabsorption. So if it comes down and binds to those cells in our collecting tubules and ducts and puts more aquaporins in the tubular cells so we can get more water reabsorbed, we have to have something that's going to draw the water and draw it towards the peritubular capillary. So the interstitial fluid has to be very highly concentrated in salt, and that way the water is going to want to move in that direction. So, and then that's how we get our wonderful concentrated urine. So if we didn't have these two processes going on, then we wouldn't have this concentration gradient where it's low here and high here, and we wouldn't have that force to get water to reabsorb whenever the antidiuretic hormone is present. And so then we'd have a lot more water being lost through the urine. We'd have to drink a lot more, or we just get dehydrated much quicker and have a lot of problems. So that is our last two objectives, or actually the last two that we're going to talk about on this video. And then I'm going to look at the case study on renal calculi, and I'll see what would be the best way to present that. So if I can do a video, I'll do that, or if there's a better way to present that, I'll let you guys know. All right, thank you.